The substance found in alcoholic beverages, known as ethanol, is a volatile, flammable, and colorless liquid. It functions as a depressant for the central nervous system and can negatively impact varying types of memory. Alcohol's effects on the brain are not uniform. Some areas are more significantly impacted than others among alcohol-induced memory impairments. Disruption of the hippocampal function is often noted due to its impact on neurotransmitters GABA and NMDA. This disturbance deteriorates long-term. Potentiation, ULTUP, which has direct links to learning and memory. Specifically, damage to hippocampal colon cells affects memory formation, with this interruption associated with the volume of alcohol consumed. High doses of alcohol notably obstruct neuronal activity in the K-one and K3 pyramidal cell layers of the hippocampus, hindering memory encoding. Additionally, alcohol strengthens the effects of GABA receptors, specifically type GABA-A, and subsequently increases neuronal hyperpolarization, which dampens the chances of an action potential occurring. Varying alcohol dosages differentially affect GABA receptor subtypes, along with impacting GABAergic neurotransmission. Moreover, alcohol at higher doses also impacts NMDA receptors, NMDARs, by reducing the ion current. This inhibition of an ion flow, normally driven by NMDAR activation, diminishes LEAD-P in the hippocampal area, with alcohol affecting LT more in immature animals as compared to mature ones. In conclusion, chronic alcohol consumption and dependence can alter the expression, properties, and functions of the GABA receptor. However, despite extensive research, alcohol's specific impact on the GABA receptor and its subtypes continues to be largely undiscovered. Alcohol consumption can significantly interfere with the functioning of one's brain, specifically in matters of motor skills and coordination due to its impact on the cerebellum. It influences thought processes and lowers inhibition and pain thresholds by casting a significant inhibitory effect on the cerebral cortex neurons. As a result of depressing nerve centers in the hypothalamus, alcohol can degrade sexual performance as well. Adding to this, the effects of alcohol on the excretion of urine through antidiuretic hormone, ADH can increase urine production. It can also negatively influence breathing and heart rate by inhibiting neuronal functions of the medulla. Highlighting on the cognitive impacts of alcohol, long-term memory, LTIM storage, which can retain the information for just a few days or even for a lifetime, gets affected profoundly due to alcohol consumption. This type of memory involves explicit, conscious, and implicit, unconscious memories. The process the LTIM undertakes, including encoding, storage, and retrieval of information, can all be obstructed by alcohol. Explicit memory, which involves the conscious effort for recalling information, including episodic and semantic memory, can get impaired due to alcohol usage. Alcohol results in a disruption in encoding episodic memories, as seen in cases of blackouts caused by an abrupt surge in blood alcohol concentration. Troubling neurons in the hippocampus. This then prevents the formation of fresh episodic memories. Under the influence of high alcohol doses, the storage process of semantic memories can get severely obstructed, specifically in relation to novel stimuli, leaving the previously learned information intact. Thus, the role of alcohol in affecting the central nervous system results in hindering semantic storage efficiency by interfering with the consolidation process from encoding. Moreover, the retrieval of explicit memory gets substantially hindered due to heavy alcohol consumption when tested against sober individuals. Alcohol causes impairments in retrieving word recognition tasks and slows down response timings in reaction time tasks. Additionally, females under the influence of alcohol score comparatively lesser in recall tasks than males. This suggests significant implications regarding the gender-differentiated impact of alcohol on the retrieval process. Short-term memory, often referred to as the magic number 7+ or -2, temporarily stores a limited amount of information, 5-9 pieces for brief periods. Any additional information leads to the displacement of previous content. Alcohol consumption proves detrimental to both cognitive processing and short-term memory due to its disruptive impacts on the brain's functioning. Studies of young alcoholic men reveal that changes in neurochemicals within the anterior cingulate relate to abnormal short-term memory function. Alcohol-dependent women researched using fMRIs show significantly lower levels of blood oxygen in their frontal and parietal regions. Predominantly in the right hemisphere, these findings align with observed short-term memory impairments traced to lesions of both the parietal lobe and the prefrontal cortex. In addition, For alcoholics, an increased volume of the third ventricle coincides with diminished memory performance. Typically tested via visual tasks, short-term memory, particularly nonverbal and spatial material, proves significantly impacted by alcohol. Visual short-term memory, such as iconic memory, receives a significant blow from alcohol, resulting in increased intrusion errors or the production of irrelevant information in delayed recall tasks. A blood alcohol concentration between 80 and 84 mg leads to more intrusion errors than those recorded within a control group. Alcoholics exhibit reduced control over such intrusion. Social drinkers, when acutely intoxicated, also display increased intrusion errors in delayed recall tasks compared to immediate free recall tasks. Furthermore, alcohol reduces both free recall, where one is asked to list given words, and discrimination task performance, thereby affecting depth perception and visual short-term memory. State-dependent learning studies in male heavy drinkers imply that intoxication during the learning process and subsequent sobriety during testing leads to a deficit in free recall tasks. This goes to reinforce the theory that alcohol primarily induces storage rather than retrieval deficits. Working memory, a crucial link between perception and controlled action, allows one to maintain and manipulate information while performing complex tasks. This memory system is believed to comprise three parts: a central executive controlling attention, a visuospatial sketchpad handling spatial information, and a phonological loop similarly managing auditory and speech-based information. This narrative primarily explores the impact alcohol has on one's working memory capabilities and how it moderates response inhibition. It's observed that alcohol leads to behavioral disinhibition, but only in those with relatively less effective working memory to begin with. Interestingly, when individuals are stimulated to perform well in memory tasks while under alcohol influence, they show improved mental scanning and reaction times, although it doesn't necessarily rectify errors. The working memory is profoundly compromised by alcohol, particularly for tasks that demand memory rehearsing strategies. On the contrary, memory tasks that don't rely on such practices are less impaired. Evidently, working memory, especially for auditory and visual sequences, is quite vulnerable to alcohol effects. For instance, musicians often battle with their performance being hampered due to alcohol, while basketball player Ron Artest admitted to heavy drinking without any discernible impact on his performance, surprising his coach as well. In terms of long-term effects, alcohol appears to create significant impact only after sustained heavy consumption over extended periods, as mild drinking does not affect cognition and may even mitigate cognitive decline risk. Yet, chronic alcoholism has been linked to diminished visual working memory and sustained attention capabilities. A striking discovery is that not only does moderate alcohol consumption during pregnancy adversely impact the child's working memory, but working memory seems to be the most critical attention aspect negatively influenced by prenatal alcohol exposure. The narrative focuses on the significant damage that alcohol can cause to the memory functions of the brain, particularly with respect to executive functions, which are closely linked to perspective memory. This impact of alcohol on memory is a theme that often appears in popular culture. There are numerous examples of characters consuming large amounts of alcohol in movies, books, and TV shows, leading to memory loss and subsequent problems. The Hangover, for instance, tells the story of three groomsmen who misplace the groom after a binge in Las Vegas and their quest to trace their steps back. With their episodic memory proving useless, a similar plot can be seen in What Happens in Vegas, where characters wake up after a drunken night to discover they are married. Contemporary songs like Katy Perry's Waking Up in Vegas and Carrie Underwood's Last Name also describe characters who cannot recall the events of the previous night due to heavy drinking. While such narratives appear to make light of alcohol-induced memory loss, legal cases such as R.V. Daviault 1994 point to the very real and serious implications of alcohol-related memory impairment.